Hello, everyone. I am Deepa Sathyaseelan, an assistant professor at the University of Oklahoma HealthSciences Center. First of all, I would like to thank the organizers for giving me this wonderful opportunity to present my work here. Today, I'll be talking about the role of necroptosis in aging and age-associated diseases. Aging is characterized by an increase in chronic inflammation, which is also known as inflammaging. In inflammaging, there will be increased levels of pro-inflammatory cytokines, in particular TNF-alpha, IL-6, and IL-1 beta in circulation. With increase in age, there will be increase in the development of several age-associated diseases such as cancer, neurodegenerative disease, metabolic diseases, and it's interesting to note that all these age-associated diseases also show a chronic inflammatory phenotype. So there is a strong association between aging, inflammation, and age-related diseases. However, we don't know what are the pathways that are responsible for inflammaging, and is inflammaging a causative factor in aging and age-associated diseases. Several factors have been proposed to contribute to inflammaging, such as cellular senescence, immunosenescence, coagulation system, oral or gut microbiota. And yet another important factor is DAMs, which is damage-associated molecular patterns. These are normal components of the cell. However, when they are released outside the cell, they can cause an inflammatory response. Few examples of DAMs are HMGB1, mitochondrial DNA, S100 proteins, ATP, etc. And these proteins have receptors on cell surfaces of innate immune cells that are macrophages and dendritic cells. And when these DAMs bind to their receptors, it will increase the production of inflammatory cytokines. In support of the role of DAMs in inflammaging is a study that shows that the levels of mitochondrial DNA, which is one of the DAMs, increases with age in circulation. As we can see here, in humans, it is increasing after age 50. The study also found that people with increased levels of mitochondrial DNA will have increased pro-inflammatory cytokines in their circulation, and people with lower mitochondrial DNA will have lower inflammatory cytokines in circulation. So what are the pathways that is releasing DAMs? We all are familiar with the well-known programmed cell death pathway, that is apoptosis. Apoptosis is associated with limited release of DAMs and therefore is a weak inducer of inflammation. In contrast, necrosis, which is an accidental form of cell death, is characterized by plasma membrane rupture. Therefore, this results in massive release of DAMs and therefore is a strong inducer of inflammation. Even though necroptosis is thought to be an accidental form of cell death, in 2005, it was identified that necrosis can also happen in a programmed form, which is known as necroptosis. TNF-alpha is one of the well-known inducers of necroptosis. TNF-alpha, when it binds to its receptor, will lead to the phosphorylation of two kinases, RIP kinase 1 and RIP kinase 3. Together, they form a complex called necrosome. The necrosome will lead to the phosphorylation of the third and the terminal kinase, which is MLKL, which is a pseudokinase. And phosphorylation of MLKL will lead to its conformational change, followed by its oligomerization and bind to the membrane. This will lead to membrane permeabilization. Once the membrane is permeabilized, the DAMs will be released, which will bind to immune cells, leading to increased inflammation. And this inflammation can act as a positive feedback loop to activate necroptosis in other cells. It is important to note that in addition to TNF-alpha, two other factors known to induce necroptosis, for example, oxidative stress and mTOR activation, is also increasing with age. So these three key factors that increases with age is also known to induce necroptosis. In 2018, we showed for the first time that necroptosis increases with age. So in our study, we used phospho-MLKL as a marker of necroptosis because this is a key step that leads to its oligomerization and membrane permeabilization, and it is one of the widely used biomarkers by other investigators. So this is the Western blot showing expression of phospho-MLKL and MLKL from the adult, that is 9-month-old, and old mice, that is the 25-month-old mice, white adipose tissue. As we can see here, there is a significant increase in the levels of phospho-MLKL and MLKL in the white adipose tissue of old mice. Then we tested the effect of dietary restriction, which is one of the strongest interventions that can increase lifespan in many species, including mice. So this dietary restriction was started at 4 months of age, and the mice we used are around 25 to 29 months of age. And to our surprise, we found that dietary restriction significantly reduced increased levels of phospho-MLKL and also MLKL in the old mice. We also looked for inflammation in the adipose tissue. So this is a PCR array data. And as we can see here, there is an increase, which is the red color shows increased expression. So compared to the adult mice, there is a significant increase in the transcript levels of pro-inflammatory cytokines and chemokines in the old mice, which was significantly reduced by dietary restriction. So this study shows that with aging, there is increase in necroptosis, which is associated with increased inflammatory condition, and dietary restriction, which reduces necroptosis, reduces expression of inflammatory cytokines. So I was showing the data for adipose tissue. Now, in the next step, we looked in the liver. Liver is also known to have increased expression of inflammatory cytokines with age, and these inflammatory cytokines are one of the reasons that can lead to liver fibrosis. The question we asked is necroptosis upregulated in the liver with age? So here we looked at 4 different age groups, 7, 12, 18, and 24 months. And as we can see here, in the earlier stages of life, that is 7 and 12 months, there is no significant increase in phospho-MLKL. However, in the later stages of life, there is a significant increase in phospho-MLKL. And that is the stage when we also find an increase in the production of pro-inflammatory cytokines. To test whether necroptosis is contributing to inflammation, we inhibited the necroptosis pathway using Necrostatin 1S, which is one of the widely used inhibitors of necroptosis pathway. Necrostatin 1S specifically binds to RIP kinase 1 and thereby blocks the downstream signaling. So we had 3 groups of mice: young mice that was given vehicle, old mice that was given either vehicle or necrostatin in drinking water for 1 month. And at the end of the experiment, we took the liver tissue, looked for necroptosis, inflammation, and fibrosis. As we can see here, the increased phospho-MLKL and MLKL expression in the old mice was significantly reduced by necrostatin treatment. Similarly, the increased transcript levels of TNF-alpha, IL-6, and IL-1 beta that were elevated in the old mice were significantly reduced by necrostatin treatment. This is the staining for fibrosis marker in the liver, which is a serious red staining. And we can see that in the old mice, there is increased staining for fibrosis, which was significantly reduced by necrostatin. And this was further confirmed by quantifying the collagen content in the liver using hydroxyproline. And as we can see that consistent with the data with the serious red staining, the increased fibrosis in the liver was reduced by necrostatin treatment. Next, we looked for necroptosis markers in the brain. And here I am showing the data from hippocampus. It's interesting that when we did the staining for phospho-MLKL, we saw the increased expression of phospho-MLKL in the old mice in a brain specific, in the specific regions of the brain, in particular, the hippocampus CA3 region. So that's why I'm showing the data here. So as compared to the young mice, old mice have increased staining for phospho-MLKL in the hippocampal CA3 region, as shown by the red color. As a control, we used the brain sections from old MLKL knockout mice and to confirm that this staining is specific. So we also went ahead and characterized which cell type is expressing phospho-MLKL. So this is data from the cortex layer 5, where also necroptosis is increased. So the first panel is a staining for phospho-MLKL. The second panel is for Innovinc, which is a marker of neuron. And we can see that there is increased colocalization in the neurons. We quantified this other cell types such as microglia and astrocytes and found that in the cortex as well as in the microglia, nearly 70% of phospho-MLKL is expressed by neurons, less than 10% by microglia, and there was no expression of phospho-MLKL by astrocytes. So here again, we ask the question, is necroptosis contributing to inflammation in the brain, which is known as neuroinflammation? For this, we first looked at the expression of the IBA1, which is a microglial marker. And as we can see here, when compared to the young mice, in the old mice, there is increased staining for IBA1, which was downregulated in the old MLKL knockout mice. Otherwise, when necroptosis is blocked, IBA1 expression is reduced. Similarly, GFAP, which is an astrocyte marker, is significantly elevated in the old wild-type mice. And when necroptosis is blocked in the old MLKL knockout mice, the expression of GFAP also went down. Next, I'm showing the data for inflammatory cytokines in the hippocampus of the young, old, and old mice treated with necrostatin. And as we can see here, in the hippocampus, the IL-6 and IL-1 beta expression were significantly upregulated in the old mice that was downregulated by necrostatin treatment for 1 month. So this shows that in the brain, as well as in the liver, blocking necroptosis can reduce inflammation, suggesting that necroptosis contributes to inflammation in these tissues with aging. Now I'm going to show a data on liver cancer, which is hepatocellular carcinoma, the primary form of liver cancer, which is also an age-associated disease. In recent years, obesity has emerged as a major risk factor for liver cancer. And in obesity, liver cancer develops through a progressive stage. That is, at first it forms NAFLD, progresses to NASH, fibrosis, and HCC. The interesting feature about the HCC development is that inflammation is a major driver for HCC development. So we asked whether necroptosis contributes to inflammation in the driving of NAFLD to HCC. To test this, we used 3 groups of mice, the wild-type mice, RIPK3 knockout, and MLKL knockout mice. At 2 months of age, these 3 groups of mice were given 3 different diets: control diet, choline-deficient low-fat diet, and choline-deficient high-fat diet. Choline-deficient high-fat diet feeding for 6 months has been shown to induce liver cancer in wild-type mice. At the end of the experimental period, we assessed changes in macrophages, inflammation, and tumor incidence. I'm not going to show any data on macrophages. I will focus only on inflammation and tumor incidence. So basically, when the mice were fed the choline-deficient high-fat diet, it will increase necroptosis, leading to the release of DAMs, which will activate the macrophages and will recruit the monocyte-derived macrophages. Together, they will induce increased inflammation that will drive HCC formation. And when necroptosis is blocked, we hypothesize that there will be reduced inflammation and reduced incidence of HCC. So this is the data showing the transcript levels of IL-6, which is one of the well-known inflammatory cytokines driving liver cancer. And we can see here that in the wild-type mice that is fed with choline-deficient high-fat diet, there is a significant increase in IL-6. And in the RIPK3 knockout mice and MLKL knockout mice, where necroptosis is blocked, the expression of IL-6 is significantly reduced. Similarly, and inconsistent with the transcript levels of IL-6, we found that the liver tumor incidence is increased in wild-type mice that is fed a choline-deficient high-fat diet. However, the liver cancer is reduced in RIPK3 and MLKL knockout mice where necroptosis is blocked. So to summarize my talk, I have shown data that aging is associated with an increase in necroptosis, as well as an increase in inflammation in the brain, as well as in the liver. And when we block necroptosis with necrostatin 1s, we could see a reduction in the inflammation in different tissues. With respect to HCC, also I have shown that HCC is associated with increased necroptosis and liver inflammation, and blocking necroptosis genetically can also reduce liver inflammation and HCC. So together, this shows that necroptosis is a major contributor to inflammation in aging and age-associated diseases. With this, I would like to thank all my lab members, especially Sabira, who is working in liver project, Nidish, who is working in the brain projects, and all the other members, and my collaborators, Dr. Arlen Richardson and Dr. Benjamin Miller, and all my funding agencies. Thank you.